Hi everyone, good morning to a less hot day, and today is going to rain. So it rains also in Barcelona for the ones outside. So I'm Oscar Camara, full professor here at UPF, and I'm delighted to present the lecture of Professor Jari Hyttinen from the Tampere University of Technology in Finland. From the Faculty of Medicine and Health Technology. He is also the head of the Computational Biophysics and Imaging Group and on the Biomeditech unit also. So he is going to give us a lecture on the integration of in-vitro and in-silico modeling for future personalized medicine. The floor is yours.
Okay, first of all, thanks, Jerome, for invitation. It's really nice being here in Barcelona, even it seems to be raining today, but nice to see people, really, not just kind of Teams bubbles on the screen. So before I actually go into the kind of, let's see the content first. So idea is a little bit to look at, as topic of today is data. So what I'm going to tell you is about what kind of data we can get from in vitro models, or what they are, and what they're doing with modeling, and how to kind of computational models can be integrated into the kind of pipelines. And I'm quite a lot of using cardiac case. There's a couple of reasons for that. First of all, I have kind of personal touch for it because my own thesis quite a long time ago was actually modeling of ischemia of an ECG. But now what we are doing a lot is really on looking the cells in vitro and how we can model those. We're doing a couple of other things, but heart and cardiomyocytes, of course, area where we have, I would say, the best models of almost any type of cells. But just a couple of words to get you kind of background of what is my group doing and where I'm belonging to. So I have this kind of group of computational biophysics and imaging. So we, as name says, do quite a lot of computation. It's biophysical modeling of cells and also three-dimensional imaging. So it's quite a lot of all kinds of tomographies. I'll have maybe one or two pictures of that. But what we are doing is cardiomyocytes. That is one area, doing quite a lot of work in neurons and astrocytes especially. That is one type of neural cell in the brain. And third type is epithelium. That is quite overlooked by modelers, but I'm not touching those today. I'm concentrating on the cardiomyocytes. And there's this BOC that is in the middle, and then we are part of one EU project and some local funding as well. And the BOC that is there is quite important on topic of today, because interesting. Kind of missing a picture. That is weird. But there was just people there, so. So I'm also part of this kind of Academy of Finland Center of Excellence in Body on Chip Research. That is totally missing. I'm missing here the picture, but you are missing the whole slide.
Maybe you can just disconnect and reconnect.
Yeah, let's try.
Yeah, the technician is coming.
Yeah.
Yeah.
It's there. But still the picture is missing, but it's my computer. So what we are doing is really 6 groups. I'll come actually back on that a little bit. And approximately 60 researchers that are doing kind of various areas of technology and biology on body-on-chip technologies. And of course, that is the background why I'm part of this, and why I'm interested on body-on-chip and in vitro technologies. And because of my background in silico, that is what I'm going to show you how to combine them. Just to kind of to highlight a couple of things that I'll tell later on the cardiac side. Of course, there are a lot of cardiac diseases, and cardiac disease are still one of the main causes of death in the world. And one interesting, not really interesting, but quite bad area is sudden cardiac death. That I think we have heard these kind of stories anyway, now and then. It's quite important. Or cause of death, also young athletes, so people can just die, drop dead. But normally there's some reason for it, and there might be a genetic disorder that causing arrhythmias. It doesn't hurt in normal daily life, but someday it just can hurt you. And of course, kind of idea that how we know which persons are prone to this, which are not. Because at least at the moment, we don't really have tools from ECG, from MR or something else to analyze who is going to die or who is not, who is in danger. To a certain degree, yes, but not really. And because whether you are going to die, it really is about your genes, but then it's about the phenotype, how your heart is built, how the cells are there, what is their function. If you are stressed or something else happened, what is remodeling the heart so that you might have this kind of really, really bad faith. So this is just one example where our knowledge, what we are knowing about disease, is not good enough. So more research is needed, and maybe the in vitro world could give some answers to that, especially combining with in-silico. And of course, one other important area of cardiac side is that this cardiac adverse effect are one of the main reasons for withdrawal of drugs. So you could have a cancer drug or some other drug, and when it goes to phase 3 trials, there might be some bad events in heart, and it's withdrawn because of this. And a huge amount of money is lost because if the drug has this effect. And now people have kind of start to look at these arrhythmia events, and really idea there is that we have different kind of assays. And of course we have a clinical evaluation that is normal, that has been done before, and then drug effects on really kind of directly on certain ion channels, for example. And then we have in vitro effects. So this is going to be part of the drug studies, to have in vitro effect of drugs on cardiomyocytes. Of course, this could be something else for other cell types as well. And actually one is in silico as well. So in silico is coming as part of the assays for drug studies. So this is kind of my view of personalized medicine. So of course we have the personal and clinical data profiles of persons that we can have. Of course it's images, it's then gene, RNA profiling, proteomics. And of course we all, I think here, agree that there's this one domain is computational models that can parse the data, can integrate the data, and through the personalization we get better predictions. But then one is as well these models that we can build from patients, induced pluripotent stem cells, and have in vitro models and future organs and body on chip as well. And of course one goal really is to get rid of the mice that is on the top, and really go from not just from mice to men, but from human to patient. And of course all this can be used, and whether all is used in this direction, it's up to, let's say, what we aim to do. But these are the data that we can, and tools that we can use. And of course I think all think here that computational modeling is important, and this is something that is my slide from my basic, let's say, lectures. So of course what we have for modeling, we have a lot of information about biology. If we think about physiological modeling of human body, we have a lot of information about biology. We have the laws of chemistry and nature, or physics and nature. And of course then we have mathematics. And when we combine it with the data from the patient, hopefully we could have this personalized or digital twin that is now one of the buzzwords. And of course, kind of traditionally idea has been used for a long time. You have imaging data from clinicians that you can combine, and that is good, and you can make personalized models. And for making models, normally if you think what data you need for that, many times you need geometry or morphology of things. You need material properties. If you are like modeling the disc of vertebrae, you need material property of these different tissues. Maybe that is available in some of the imaging modalities. But then in maybe you might need also some really functional data of the cells. Of course, it's available somewhere in literature. But if you can really get patient data from that, that might be one way step forward. And that, then we can come to the area of in vitro models. So, anyway, the preclinical animal population data that has been available for a long time, and now we have the emerging in vitro models. And just to kind of stress that this is just one way of modeling. It's like the same that we have the in-silico models. We know that every time you make a model, it's a limitation. It's modeling for a certain aim. As yesterday, I think in a couple of lectures, there was really concentration or idea that you need to have a need for the model. What is used for the model, and you start building your model based on that. And similarly, if you think about these in vitro models, they are as well similar models. They are quite limited view, in a way, of the human physiology. They are lacking a lot of things, but similarly, the computational models are lacking certain things. They are just for a certain purpose. Similarly, in vitro systems are doing that, the same thing. And really, the question is whether it has those features that you are going to model that are needed for your information that you have in your research quest. And I think we all know this already, but of course the kind of what has changed in vitro is the Professor Yamanaka's Nobel Prize on how to make stem cell models, stem cells from any of us. So introducing the genes for your cells, whether they are skin cells, your blood cells, whatever. If you introduce the genes that define the stemness of the cell, we can actually generate stem cells that you can then generate and change to any kind of other cells of the human body. Of course, why this is there is of course really important. We can make the models of all of us. Of course, it still needs a lot of money, but it really enables to have a source of cells. You don't need to get piece of your heart or piece of your brain and put it on the dish and test certain things there, but you can really grow part of your heart or part of your brain on dish. And for example, test drugs on your cells and see what is happening. And to a certain degree, this has been done, and a lot of work is done towards this kind of direction. And for example, in Tampere, what we have had done quite a lot is on using this kind of in vitro tools for analyzing cells. These are cardiomyocytes beating, and on top there is microelectroarray. So that is a system where you grow your cells on top of electrodes. It's like a dish with ECG electrodes on bottom, or EEG, or whatever cells you put there, and you can get the signal from the electrophysiology. By the way, those were not iPS cells because they were not invented in 2007. Those were actually human embryonic stem cells that we used to start with. From embryos that were kind of used for fertilization, but they were not so bad, not so good that they were really used for fertilization. They were going to rubbish bin, so the stem cells were used for growing heart and brain, other cells. But now everything is iPS. And so what these kind of body-on-chip systems are, because of course this is where we can have cells on top of electrodes, but there's just cardiomyocytes there. If you like to build like a tissue, we might have more like interaction of cells. And now you can buy from the shop. There are a couple of companies building these kind of ships where you can grow, for example, 2-3 different cell lines in different compartments and have their interaction through microfluidics. For example, this is one example where they have actually part of the brain. So there's astrocytes, pericytes, endothelial cells, and looking how the endothelia, that is the blood vessel part, and then the brain, that are astrocytes and pericytes, that are important for controlling the throughput of blood-brain barrier. So this is a model of blood-brain barrier on a dish. And there's quite a lot of these kind of relatively simple microfluidic devices where you can grow cells, look in their interactions, and more are coming. It's actually a new word for this kind of devices as well. They're microphysiological systems. That's another word for that. Next week there's a conference in New Orleans on microphysiological systems. But if you really look at the kind of how tissues are structured, they are of course not just single cell types. They are multiple cell, like it was on previous. This is a picture from heart actually showing the how the blood vessels are kind of really lining almost every structure of the heart muscle. And they are not just cardiomyocytes, there are other cell types as well. So we are still quite far from having real tissue, because tissues have a lot of cell types, a lot of functionality, vasculature, nerve cells and others. So is there actually any way I can see the log? Maybe you can tell me when I'm not a single piece of time machine around me. So how we can make better in vitro models? Of course, one important thing is that we would like to make them more like real tissue. And to look at tissue interactions, because heart is not alone in the body, there's other cells as well, other organs. And really big problem is how to make them more repetitive and defined. And of course, one big thing is from 2D to 3D, because anyway, our organs don't grow in glass or plastic, but they really have an environment of the cells. And of course, the next line is how to keep them still simple. In a way, just like our computational models. If you try to build a model of human, you cannot have all that complexity in your computer. Similarly, if you try to build on dish, then you kind of lose part of the simplicity of handling these stuff. And just to kind of give an idea that what we are doing in Tampere is that we have these 6 groups working together, so 3 engineering groups, myself, then one from micro nanosystem fluidistics, biomaterial, and then we have people from heart and nerves and of adult stem cells that are coming from harvesting different cells from human body. It's bone marrow or fat. But just kind of the components of these body on chips. Of course, first we need the cells and to grow. They can be also harvested from the human, but quite rare. But these induced pluripotent stem cells is one most important source. Then there's really the microfluidics to steer the control the environment. They are fluidics, but they are also oxygen and other areas. Then we have methods to analyze the cells. So whatever is your aim for looking, whether you're looking for cardiomyocytes, for neural cells, you look to electrophysiology, maybe you look for the mechanics, you would like to see the kind of gene profiling, calcium analysis, whatever, you need tools for that. And of course we need the materials where the cells grow. And I would claim that this one corner here is computational modeling that is kind of going to be part of that. It's not very common to have that, but it's I think very important part of that. So how to kind of look into the what the cells are doing, to understand the cell responses. If you are stimulating the cells, you see what is happening. Maybe if you build the computational model, you can understand more the mechanics, what is there. Of course, there's a lot of modeling on the environment, for example, but that's a kind of different modeling than going for the models of the biology. Just to kind of, what is really nice about these, that unlike in a human body or animal, certain degree you can do in animal, but you can have like input-output relationship. So we can have, like here is one, we can inject certain things in drugs or even genes in single cells. We can have the electrical stimulation recording. We can have mechanical stimulation of the cells. This is heat profile actually. We can have heat or something else, physics, microfluidic, shear flow, shear stress, like here. And through this, of course, then we can have different measures to measure it, what is happening from the cells, whether we measuring the environment, whether we measuring the cell functions and so on. So we could kind of think that the in vitro models, if they are especially from certain kind of patient, we can have the genotypical model of certain person or certain type of persons, or we can have really personal model. And when we have the control and input system, we can control the environment. So we could maybe change the flow, we can change the oxygen, so we can have a disease. We could generate trauma, mechanical trauma or something, environmental trauma, put some drugs, put some bacteria, whatever you invent. And that's generate us different phenotypes. If you grow the cells with different environment, you might get different phenotypes. And if you stress the cells, you could get different kind of responses and functions. And of course, then the question is in silico. Can we dig into the mechanics of the biology so that we can understand what is happening there? And one maybe question is, can we use in silico to translate to clinical? So let's look at the cardiomyocyte a little bit on this. And of course, as any system, whether you look cardiomyocytes or your vertebrae or something else, this is the problem on physiological modeling. You have really different kind of scales from the proteomics to kind of cell machinery, like here's the myosin acting complexes, the cells, the tissue, and of course the whole organ. This is the rat heart, actually imaged by micro-CT. So how to handle this all? And of course, it's this normal paradigm that organism is affecting the genes, and genes are dictating how the cells and other work. So we have really this kind of, it's a multi-scale and multi-level interaction problem. And normally what you are building your models is normally in some of those domains. You are looking at the interaction in genes, or you're looking at the organ level, and now more and more interaction on these. Of course, in vitro world is mostly on cells. We can have some kind of tissues, not real tissues, but certain kind of tissues, and then we can look at the mechanistics. And let's hope you have. I haven't actually looked what you have seen here. That if it looks weird, just say me. Now it seems to be okay, but mainly look at my own screen. So if you're missing something there, I'm talking something that you don't see. Just let me know. Okay, so we have kind of in a way worlds of kind of clinical side and modeling of the whole heart and thorax, and then we have the cells in vitro, and of course this is a huge gap. I'm coming back to that, but of course the question is, can this iPS-derived cells offer actually some personalization of the clinical world? That could be something, but it's not really done properly at the moment. People are doing really nice model of the heart and from clinical images, and of course the cell models that you can see in a moment. But this could be something interesting. And of course, why this is interesting as well, that if we think that we could kind of make these cells in vitro to have similar kind of environment, kind of problems as in real heart, stress, oxygen demand, or something else, we could actually see how the cells are deforming, how the tissues are remodeling itself, and getting back the information to the whole heart. there's actually modeling kind of, I would say, problem as well, that these worlds are quite far apart from kind of how we model them. Another one is kind of continuum models of so-called bidomain or Laplace-Poisson equation in electronics. Another part is kind of point models of the compartments of different cardiac system. And to my understanding, I think we still miss parts here that we could really translate these, but I'll come back to that later. No point, no. What is the time actually? Just to get some reference.
You're doing 10.
10. So I have 1 hour. Nice.
45.
45 minutes. Yeah, so some minutes for them. Yeah, let's try to fix that. Okay. So if you think about what we are doing in the computational models of these cells, of course, some of you, of course, might know it, but it's so-called Hodgkin-Huxley models that was built already in 50s. Hodgkin-Huxley got the Nobel Prize of that, and it's kind of simple model where you have a membrane model. Then over the membrane, of course, what happens is that it's some passive electric charges going through it. It's really an insulator. And it's making like a capacitance between the inner and outer part. And then they have conductances, that the conductances are emerged from the different ion passing through the membrane. And then there's the potential that are driving each of these ions through the membrane. Looks simple, but each of these conductances are really complex because they are ion channels that are opening and closing. And you have the dynamics of opening and closing, and they are nonlinear and also depending on each other. So the model actually becomes quite complex, even in a single cell level. But of course, on below here, you can see the kind of cardiac muscle, and you have the action potential, and that is the action potential that is really driving the function. So it's a fast upstroke, and there's a lot of plateau and going down. And what that is driving is the calcium event in heart cells. And that calcium is then driving the contraction of the heart. And there's these two pictures here. So where we get data from these is, of course, one important is so-called patch clamping, where you can really put a pipette electrode on the cell surface and measure maybe even individual current of ion channel and determine these opening and closing. Or then we have this kind of heart in-vitro electroarrays where you can measure, it's more like ECG. But people have done a lot of work on defining these ion channels. And of course, I think many of you are doing this musculoskeletal world on looking the contraction. And of course, the mechanistics here is exactly the same. You have the, let's say, the myosin and actin working together. And of course, modeling here is important as well. Surprisingly, not so much done in cardiac field. People have really been enthusiastic about ECG and modeling that. But of course, this is becoming more and more important. To look how this machinery works. And why it's important to actually look these is that if you think cardiac diseases, it can happen, of course, in general metabolics, but it's affecting other cells. But if it's about electrophysiology, it's pinpointing to the ion channels. Something is going wrong with these conductivities, and then the function will become different. But it could be here as well. A gene, mutation, or drug, or something else that actually changed the machinery, molecular machinery of contraction. And of course, if you are modeling the whole heart, you can do a lot of work with kind of clinical world there and simulating patient others. But if you look really kind of mechanistic view of the heart diseases, then you should actually have a look on here. And of course, below this is the gene machinery and RNAs and other stuff that is causing these stuff. Okay, just go a little bit back to the in vitro. So this is where we started, and we were really excited to have human cells, and then we had different kind of drugs, and we could easily see the drug effects on vitro. So this was nice. And of course, these were not iPS cells. We were not really sure what are these persons, because we were not allowed to actually know who these persons are, because they were garbage-going embryos, and there was no knowledge about their background. But then came the iPS, and what was kind of nice that us and many others showed that, of course, the iPS cells taken from the patient actually retain the genetic machinery, so it retains also the diseases. So this so-called long QT phenotype, so in a healthy patient, the action potential looks like this. And in ECG, you can see the effect of here the. Let's see how I can. Here I can see. So you can see that the action potential is pretty short or normal, and there's what happens here, that this Curas and T complexes here for the relaxation. This is what it should be, but in these patients with long QT, this is longer. And there's what is causing this is certain change of ion channels for these patients. And if you have a normal patient, it's look like this, but when you have a sick patient and we have the cells in vitro, we can see the same thing that is happening in the patient heart. So kind of showing that we can do it in vitro. Have a really patient with disease in vitro. By the way, it's kind of nice, kind of small story here that Katrina Aaltosetälä, who is a cardiologist and our head of the cardiac kind of heart group, who is actually deriving these cells, he was actually in UCL, California, when Professor Yamanaka was there inventing the iPS. So we were really early to adopt these iPS, and this is one of my papers. I'm part of this that has most citations, and reason for it, Professor Yamanaka is part of this. So just if you would like to have a paper with a lot of citations, try to write it with the Nobel Prize winner. So. Anyway, my part having this kind of Yamanaka there was kind of, I was not involved in inviting him, but Professor Katrina Aaltosetälä really knew Professor Yamanaka. But of course, this is kind of idea that we have unlimited source of cells, because we can renew these iPS, we can generate a lot of new cells, and whether it's a patient cells or healthy or different gene, it's an individual with different genotype. And hopefully we can have the phenotypes as well in vitro. I'll talk about that already. But in a way, if you think, let's go back a little bit. If you think that you need to have this kind of measures where you batch the cells, it's art form of cells. It's really painstaking work. Or if you grow these cells in this kind of system, analyzing these is a lot of work, and having these electrorays is quite costly. A couple of €100 per one of these. I think now they are a little bit cheaper, but still. So how to get really high throughput, how we can get more diseases and multiple functional biomarkers, maybe not just electrophysiology. We would like to have really tissues, not just cells, and insight on the disease. And the translation is, these are all still kind of missing, more or less. Of course, you could kind of use these techniques to, and actually Professor Aaltosetälä has treated some patients so that she had cells on vitro, tested a couple of drugs that was used for this kind of syndrome, and selected the drug for that particular patient. So it can be done, but it's not really part of clinical routines. But this is kind of wish list, what we would like to do. And now actually looking these cells, we started quite many years ago already. Quite simple image processing, and you can actually get the mechanical beating of these cells. And of course, this is much easier because you can grow then cells upon any surface that allows microscopy and video, make the video, and then track the cell beating and have the curves. And of course, it's actually nicer than electrophysiology, because this is what the heart should do, is pump blood. And this is kind of mechanical force now, or the mechanical function of the heart cells in vitro that we can follow. And we went further, actually, and there's a lot of optical methods where we can actually have calcium imaging there. Those have been around for a long time, and now you can have the calcium indicators coded in a gene. But there are also other indicators that you can do. And you can have all these sensitive dyes, for example. And this is one paper where we show that you can actually have all three simultaneously. We can, with some image analysis tricks, some imaging tricks, we can simultaneously measure all these calcium voltage and the contraction from cells. So now we have actually quite nicely figured out how to get the machinery out of these, so we can really have all three curves simultaneously from the cells. And it works in certain diseases as well. So there's a definite change of beating, for example, long QT. It's not just actually making it longer, the QT time, but it's actually, it's quite a marked change. It's actually more drastic in the single cells than in the heart tissue. So we can really quantify diseases by using this method as well. And lately, in this Center of Excellence, we have built systems, or Professor Pasi Kallio's group is building a system for controlling oxygen levels. And for example, here is oxygen concentration on dish, and then it's dropped down and up again, and we can see the kind of how the beating rate changes, how the activity times change on these cells, and how the electrophysiology change. So in principle, we have shown that we can have these patient cells. We can model diseases, whether diseases is from kind of inherited disease or it's from the, let's say, environment, or the like ischemia. And we can have drug effects. And so, in a way, we have the kind of modeling platform for many cardiac diseases in vitro. But there's still kind of problems, because the heart cells, this is not typical, but this is kind of one example of heart cell beating. As if you compare that to this is adult cell, they have really nice elongated cells that are kind of communicating to the ends, the neighboring cells. And of course, these cardiomyocytes, if they are single cells, they look like amoebas or just blobs there in the. So they lack a lot of the systems that are in real cardiomyocytes. And of course, that is the problem. And because these are the models, so on in vitro. So we wanted actually to understand more of these and build in silico models of these same cells. It's kind of building model of the model. But still, because these are the things that we can get all this information, it's nice to have a model to understand what is going on in vitro. And just motivating why we are doing it. And if you have these cells in your computer, you understand, maybe you can transfer that then to, let's say, more adult types of cells or something else. This just shows actually one problem is that these are gene expression of certain genes. I don't recall anymore. I think, yeah, they're sodium, calcium, HERK, and IK channel genes. And they are adult, and the eye cells and core for you are two different cells that you can buy from shop nowadays. And you can see that they are different. Of course, these are individuals where the cells have been derived. So they could be changes on the phenotype, but there's also change on the of the phenotype, because if you get data from one lab to another one, they might be different. Because the environment are a little bit different, the phenotype is different in another lab. So, but it's the same thing like human heart. If you take one heart compared to another one, if you have a lazy guy or top athlete, it's going to be different. Anyhow. But in single cell level, it's even a bit bigger. So we have shown that you can have higher throughput, you can have diseases, multiple biomarkers, but how about mechanistic insight into disease and translation? Maybe the in-silico would work. So finally I go to in-silico. A lot of a lot of work in vitro. And this is actually a slide made by Jussi Koivumäki, who is a kind of lecturer now in Tampere and part of my group. An idea really is that, I put my mouse there. So what you have now in vivo from patient, of course you have MRI, maybe contraction data from MRI, ECG, and based on that we can have this 3D model of the person. And in vitro, we can get same patient, we can have the, of course, protein expression, we can have the, let's say, the in vitro model on the iPS heart and get the ion currents, action potential, contraction, and so on, calcium. All this is available from in vitro and this from the patient. And if we can make this in silico model of these cells that are in dish, of course we can look how these ion channels are there, how we should modulate it so that it resembles the patient cell. And maybe then we can translate that to adult cardiomyocytes and put those cells in the real heart. So this is just one piece of puzzle on this puzzle. And of course, this is what these in-silico cardiomyocytes are. There are a lot of equations for each of these ion channels. You remember the earlier model of the three conductances. Of course, each of the major conductances, different ions, are coming from multiple different pumps, multiple different exchangers, and all of these need equations and parameters. So there's a lot of parameters, a lot of equations, and just this is the just an example, one equation for this sodium-calcium exchange, and each of these points here or bumps have similar kind of equation. And of course, based on this, you can start modeling the electrophysiology of cardiomyocytes. And the data where these come from is that some people have done a lot of work on identifying by patch clamp. So we have this kind of curves on each of the ion channel, the opening and closing dynamics. And these are transferred through this equation and parameters to a model. So in a way, kind of traditionally, it was really a lot of pain to do this. And there were, of course, adult cells doing it. People have done it from dog, for rabbit, then human, but human data was really scarce. But now we have a lot of these iPS cells, and these have emerged. And I think my lab has really pioneered on this, especially Michel Aselopaki, who was postdoc in my lab for years. But with this data, of course, we can have really all ion channels, all ion currents, and individually look how they are in these iPS cells in silico. And this is just an example that we could have like a long QT syndrome. That was the first one we analyzed. And how you can do it in these cells, of course, is that you make the normal cell, then we know that there's a problem in this ion channel. You can change these parameters of this ion channel, and then see whether you actually generate the phenotype. And of course, then we can compare that, whether that model is fitting to those ones that we can measure in vitro. But of course, in a way, this is quite limited way of doing things. So what has been lately, I think, in all modeling world, has changed so that, in a way, we have always problem of uncertainty of data. Whether you are talking about cardiomyocytes, whether you talk about your vertebrae or something else. There's certain range of measured data from individuals, from let's say, there's errors, there might be some phenotypical or genotypical changes. How we deal with this. And there's a couple of methods to do it, and one way of doing it is making populations. So instead of having single cell, you start varying your data, your parameter data, and build huge population of models, tens of thousands, hundreds of thousands of your whatever you are modeling. In our case, it was cardiomyocytes. And then those are the action potential generated by the cells. So we have huge number of cells, each generating a little bit different kind of phenotype or genotype. So that's the basic idea. Build the kind of brute force. Do a lot of models. But of course, not all models might be really good. How you do that? So how we did and many others are doing is that we have measured data. Those are actually those are different biomarkers from the action potential, like peak and width and so on. And each of those boxes represent a measured set of data from different labs. So each colorful box is a data sets from different labs. And then what we did, we selected only those models that fit inside these data boxes, measured data. And those that are outside this measured real cardiomyocytes in vitro models, we tossed away. So only those that fit all these parameters are in the measured kind of area, we use those. So we get rid of all those that are gray, and the black ones are what we used. And when you start using this, there's one example of we could try now in-silico drug test. So this is a one channel blocker, and what we see that it kind of here it behaves on these cardiomyocytes as usually. Then some of the cardiomyocytes actually produce arrhythmia, and there's even cardiac arrest. Of course, this is happening in computers, so no patient have been harmed or no cells have been harmed. But of course, now we can look what are the properties of these, like these guys here. Is there something we know about these that we can go back to the bench and test them in vitro? Maybe even looking at the clinical data. We haven't done that kind of analysis yet, but and of course, similar thing we can do on these populations. We can have population of normal persons, population of like here long QT, population maybe some other disease, and then for example having this one drug, and see how it affects. And this is a different doses, and we can see the dose response on these different mutations. And it's quite interesting actually that first of all that some of the title is missing from you and also in my screen, but I'll explain. So what is here is that when we now look at that population of models that has the long QT gene, so that has the elongated kind of change of the ion channel, we actually noticed that some cells, even there was this kind of a change on ion channel, actually behaved normally, and some really had the phenotype of long QT. And then when you actually add the drug to these cells, they had a different response. So those cells that actually have the mutation, that didn't have the phenotype, didn't have the long QT, actually didn't respond to drug, but those cells that had the disease and had the phenotype actually responded to drug. We still need to really go back to bench and see whether this is really happening in vitro as well. But this is kind of interesting way that we could kind of predict something in silico. And it's to be seen that whether it's real observation. But lately what we have done, kind of wrap up backwards, is that we have actually used this kind of optical data, not electrophysiological data from electrodes, but optical data from calcium and from the electrophysiological optical data, and tune these kind of population based on that. So we can have the population, and we can tune the population so that it matches to certain kind of data in vitro. And if we have a normal population in vitro, we can tune the population to match that. If we have some disease, we can take another part of the population. It's kind of making the modeling much faster. We don't need to generate any more models individually for certain kind of type of patient. We can select them from the library of the population. And just to show that what we are doing lately is to go for not just electrophysiology, but also for the cardiomechanics. And this is actually a paper that you can find online, but it's not yet published, it's under review. So we actually combined here the electrophysiology model, then the actually RISE model of the contraction that really pinpoints the kind of, let's say, the dynamics of different on the molecular machinery of the myosin and action system, and also the energetics. So how the ATP is available, how it's consumed in the contraction process, and actually shown that it actually can explain, at least in theory, quite a lot of that has been seen in drug effects that have been now coming into market. But as I said, this is not reviewed yet. It's under review. But just to kind of show, this is not just our data, but there's a lot of data from the literature on different labs that was collected for this patient, for this model. And this was, all this data was really collected first to tune the parameters, to tune the models, and then to validate. So we applied the drug with the model and see how it performed compared to in vitro data. So you can make the models, one kind of lesson to learn here, you can make the models, but having still in vitro data is a pain. And your own lab or your collaborator's lab might not be able to really produce that. So like here, we really look at the literature and try to harvest everything possible. But in a way, this is kind of, now that we have the models, we have shown that you can tune the models, make the models in vitro based on let's say, the in vitro data, whether it's electrophysiological data, whether it's this kind of data from the optical measures. We can tune the populations, do the drug testing, do certain kind of disease modeling in silico. And hopefully, of course, then go back and select certain populations, certain kind of condition that as predicted by the models, and then go to in vitro. And of course, then hopefully these in vitro, we can really have a patient-specific models, or they have, and we can help the patients as well. That is the hope in the future. Okay, but there's still kind of one thing that is not there. We have the cells there, we can see how the cells work, but of course it's not really very good cardiomyocytes. This is actually very nice heart, as you can see, beating. That is from this Professor Katrina Aalto-Setelä's, actually you can see it on their webpage. It's just accident that they stem cell generated heart. It's in 2D disk, so it's not really a heart, but it's just nice shape. But we still miss these real kind of in vitro systems. And really the structure of the tissues are not really developed in vitro. And what we are doing, and many others are doing that as well, that it's not really great video, but it's not actually functioning. Sorry about that. But on the middle, it's actually still unpublished data, but what we are trying to do is to look at the 3D cell cultures, and for example here to analyze the contraction on 3D. It needs some tricks on microscopy, it needs some tricks on analysis, but we have shown that in principle it can be done, certain limitation from these 3D cell cultures as well in cardiomyocytes. And there's actually quite recent paper published in Nature Biomical Engineering from a group of people. Some modelers might find some name from the Simula, Oslo, and from USA. What they have done is trying to find out ways to make more like mature cells. And how they have done it, they have kind of mechanical environment, but they also have this kind of metabolic environment. And that is making the cells more like mature alike. It's interesting. Normally what people are doing in cell cultures, that you are actually putting some food for the cells. And normally it's sugars. There's a lot of sugar in the cell culture systems. And it's for every cell type. But only thing that they have done, actually it's done before as well, is to change that to fatty acids. That is more like in real tissues. It might work in other cell types as well. Who knows? But kind of showing that what we do in vitro, we have quite a lot of learn how to really make good tissues, and even small things change how the cells survive. But it's a nice paper. If somebody's interested in this, please have a read, because they have also actually used Michelangelo Spacchi's old model that's published, and then use that to simulate these cells and mature the Michelangelo's iPS-derived cardiomyocyte model towards adult type, and show that when it's certain handling of calcium especially is changed to an adult type, it matches to the T cells. And of course, another side of the story is, if you think about the heart and modeling of the patient, is that now there's actually quite a lot of papers emerging how to make patient-specific heart simulation, whether it's atria or ventricle or something else, really take into account the kind of all the information you can get from the ECGs, from the MRI, different techniques, and combine that towards modeling on that domain. So in a way, there's kind of maybe in someday we could actually have this kind of integration of this in vitro world. Oh, there's still one missing. Ah, this is weird. Okay, one video was missing, sorry. So that really kind of having these steps that we can do in vitro, maybe better cardiomyocytes, better tissues, and have them also integration better to the models of the patient in let's say, that is to analyze what is really happening in a way, in a clinical situation. But of course, it's not something we need to do for every patient. It's maybe something we might need for certain cases, but not very often. But just to kind of, this idea to bring these gaps together is important. Just to show a couple of these last year's pictures, but still we actually have cells now in vitro in Tampere where we can have vasculature in heart, but also some other tissues. And we have also innervation inside these tissues. So they are not mixed yet, but we hope to gain in a couple of years really cardiac tissue, some liver, some other tissues that really have integrated vasculature and innervation, and we can have look in the interaction of these cells in vitro environment. And of course, hopefully build this in silico in the same phase as well. But this really this in silico modeling world is really expanding a lot. I remember when we actually applied this, our grant from Academy of Finland for long and big grant for this body on chip research. At that time, 5 years ago, there were maybe a handful of papers, but now there's a lot coming from really different kind of organs and integration of different tissues, different organs together. And as I said, there'll be next week this World Symposium or Summit, actually, World Summit on microphysiological system. Let's see. Hope to be there as well. But if you think like future precision medicine domains, of course, there's all these, whether you go for genes, RNAs, and there's a lot of happening in imaging. you can think about mobile health and stuff like that. And of course, this is something we always claim that we integrate, and computational modeling are part of that integration. But of course, the in vitro body on chip and computational model, I think those going to be part of this whole business. How much they will be on the basic research that is going to be now, I'm quite sure there'll be big role in drug studies, certain understanding of diseases. How much those are really part of clinical pipeline, I doubt, but maybe in certain cases, yes. Okay, and of course, it's not me who have done this work, it's mostly others. So again, there's a picture of my lab missing. For some reason, this doesn't like to show faces of people. The body on chip research collaboration picture was gone, and my lab picture was gone. Maybe it's really tuned to in vitro. So. But especially these guys there on understanding the cardiomyocytes, Jussi Koivumäki, Michelangelo Paki has done a lot of work on these, really pioneering on iPS-derived cardiomyocytes. And lately, Antti Ahola was the guy who has done all the video analysis. And Amin is now doing the work towards the biomechanics and energetics. But also people doing this imaging has been there, and of course the collaborators, especially Professor Katrina Aaltosetälä, who is our cardiac specialist and head of the cardiac group. And we are organizing computing cardiology in Tampere in autumn if somebody is interested to come. I guess the deadlines are gone, but the conference will be there. So welcome, and thanks for listening.
Very good. Thank you very much for the lovely talk. I mean, it's just crazy that you have answered all my questions that I had through the lecture. Any questions from the audience?
Nice.
Benjamin Gantenbein, University of Bern, Switzerland. Very nice talk. Thank you very much. How do you think can you cover the donor variation when I think you deal also with primary cells mainly in your chips, right? So how do you deal with the variation, and how can you generate many replicates and then deal with the optimization of the cost? Because you told something that one of your devices was also several hundreds of euros, right?
Was there more than one question? There's one is cost and one is variability, I guess. So I think if we talk about variability, of course, this if you take you talk about primary cells. So if you really take a patient's cells and put it in vitro, then of course because they are really coming from the heart or other cell type that has been in organ, it's been matured, it's been kind of functionalized to that purpose in your body for a long time. So the variability of those cells is less than those that you drive from iPS-derived cells. And so those are better source, but of course primary cells are not so often available. Of course nowadays more and more. And of course there the problem is that how long you can use those cells in vitro. Because when you take cells out of the body, they start to change. They are not anymore in the environment, they don't have any more the loading, and they start to change. So even adult cells start to kind of, let's say, go back to, let's say, really, or reduce their functionality as they are compared to body. And iPS cells, it really is how you standardize the way you cultivate cells and how you standardize the environment, how you make it so that it mimics more the real let's say, the cell environment in the body. Actually, I saw one paper telling that your cardiomyocytes are ready when you are 7 years old. So it takes 7 years for your heart to mature. So it's no point that we can ever have 7 years time to grow cells in vitro. But of course we can try to make them better, more like adult-like. And regarding the cost, and I guess the certain, if you need to go to this kind of really detailed level of measurements like patch clamping, that's huge in cost, because it's not that the equipment, it's the person time who really patches a couple of cells per day. If you have if you have the microarray, they really are a couple of hundreds, but they are reusable, so you can use them. And that's why we went for the optical measures, because then you don't need, you can have a normal cell culture systems. The challenge, actually now going actually beyond your question, is when you have these chips, you have multiple cell types, how you image and how you make the online measurements of those. And it's actually quite rare that you have that kind of systems at the moment.
More questions?
Ah, good.
Thank you, Jari, for the great talk. A bit, so I'm just sizing the opportunity of this discussion about to ask a question about the experimental setups, so microfluidic setups. So then talking about the behavior of the cell in a real tissue environment. So what do you think about these mesoscale microfluidic devices where you have actually the cell embedded in an extracellular matrix within the microfluidic devices? It's being increasingly used in tissue engineering and in cancer modeling. Do you see for cardiomyocytes, do you see it as a valid model?
Yeah, of course, it's again, it's what you aim for for your modeling, whether it's in silico or in vitro. And of course, we are trying to go that direction as well on having in cardiomyocytes, but other cell types as well, what we are doing on really kind of having 3D with excellular matrix that is either produced by the cells or mimicking the cell environment. Of course, there's a lot of challenges and imaging, a lot of challenges on if you would like to have online assessment, what is happening there. And of course, another one that we are kind of looking forward on that as well is to how you control the environment in real 3D, because it's very difficult to make really vasculature. If you can make vasculature, can you actually have something like blood? I guess nobody have had that. So how to carry the oxygen to this system in 3D and how to control the oxygen environment and maybe other nutrients as well in 3D. I think that those are really, really problems on these 3D environments.
That's where modeling can definitely help them.
You need the modeling really on environment as well.
Just related to this, I don't know if maybe it's really out of the blue, but do you see a potential relation with the most advanced 3D printing technologies and the relation with this body on a chip?
Definitely, people are also using 3D printing. Actually, us as on the certain level, I think now 3D printing has been done quite a lot of on building this kind of tissues for let's say cell replacement therapies, and of course they are available also for building these kind of environments as well for these chips. Definitely.
So something that I really like it is your attempts to really incorporate contraction. No, because in this field, it just looks like it's cellular electrophysiological models, and there is no movement there, and there is nothing in terms of the contraction. Have you seen really differences in phenotyping when adding the contraction model with respect to only having the electrophysiological part?
In principle, yes. We don't have we have certain diseases where we don't really see a lot of changes on electrophysiology, and you can see the changes on the contraction, because some of the diseases are actually, for example, HCM related diseases, or this hypertrophy of cardiomyocyte, cardiopathy related disease actually affecting either the calcium handling or then the actual machinery of contraction. And if you have something there, of course, it doesn't really show off in electrophysiology. It might show off in ECG to a certain degree, but of course it might show off in cardiac output or something else much more than ECG. And same thing happened on vitro models as well. So definitely the contraction is important in certain disease types, especially.
And another important concept is the role of this body on a chip on the in-silico trials, and as a part of validation of the models, right? And
Sure.
part of the VMV40, the verification and validation, because mainly what you have presented is the use of the models to some extent incorporate the information of the body on a chip, but not really for the validation, right? How do you see the body on a chip in the whole pipeline of the validation and also a role in the in-silico trials?
Yeah, I think, of course, the kind of what we have done, we tried to build the models to kind of understand the biology a little bit. And of course, part of that will be as well validation, and we use the in vitro data for that. But another way, of course, there's now we have real cells, we have kind of real biology. And if you're building that kind of, a model for let's say, other purposes, and you try to kind of validate your computational models, of course, the in vitro is a nice environment because, as I said, you can control it. You can have this input-output relationship that you cannot have in human or animals. But of course, I think that might be even an area people have overlooked, because you might need to build these devices and systems towards that goal as well.
Is there any of these kind of systems already used by the certification agencies? I mean, for FDA, etc. The
CIPA initiative in USA that is having also this in vitro system, and I'm not now 100% sure that is it really officially there, but that is the kind of really proposal to us, this kind of arrhythmia drug evaluation. And but I guess there are no really standardized methods either.
Yeah, I think that's a big issue because there is no initiative which is called C4 Bio, as trying actually to replicate even simple material tests. Well, simple not, because with avatars of biological materials and avatars thereof. And they're seeing that even by taking the synthetic avatar, multiple lab experiments give variations over nearly 100%. So that's really a challenge to standardize this. And when I'm thinking about these single cell experiments might be very sensitive, that's an issue. And just to comment on the VNV40 stuff, this is basically this kind of attempt for regulation came out really macroscopic simulations out of engineering for medical devices. And I think that when we dig into biology, this kind of modeling, the paradigm has to be a little bit revisited, and probably talking more about good simulation practices, try to ensure robustness of the modeling, maybe consistency of different hypotheses, consistency checks, which is indeed not really considered in the traditional engineering view of verification and validation of models.
Yeah,
so
I think it's many of the people who are doing this in vitro work or stem cell work, they have the same disease as most of us. So when you have a model, you would like to make a better one, a new one, and having the idea of building the standardized and robust system is not kind of part of the genes of the people who are studying this. So.
I kind of disagree with you, Jerome, but as you are the organizer, I don't know if I'm allowed to. But yeah, I know. But in the end, the VMV40, I think it's applicable to whatever. I mean, to whatever computational technique, because basically what it's telling you is just that you need to be sure that you know what your model is for. And that's the main principle. It's like you need to define the question of interest, the context of use, on what you are going to use your model, which is the data you are going to use for validation and verification, and which are the risks of a bad simulation result. And this is valid even for AI. I mean, for any computational technique, you need to really know this kind of question of interest and context of use. So I don't really see, I mean, maybe, obviously, the verification procedure will be obviously different, and the data will be different. But the main concept, to me, is valid independently on the scale and even to other computational techniques. On AI, this is one of the things that are really missing. I mean, this concept of verification, validation, and defining the limits of what you are developing.
Yeah, it definitely deserves a full roundtable. We can think about it for next year. But there are a lot of very important points in what you've said. So the concept and the structure of the concept is right and is certainly almost universal. These are the practical steps you need to implement in order to achieve standardization, etc. But that's maybe we don't have time to develop that right now. I have few technical questions, but we're only the two of us talking here.
No questions from the audience? Go ahead for the technical.
Looking for the coffee? Or?
Yeah. No, it's about this concept of population modeling in cells. So now, with this in-silico cohort, etc., we're talking about population modeling of patients, etc. It's kind of simpler, no? And that's simpler in the sense that it's very clear. So we are all different from each other, and we will try then to grab this diversity as much as we can. Now, and I have the impression that in the kind of population modeling you're targeting, so by taking information from single cell, there is an added complexity because I'm wondering within the heart, so you have the cells, collection of cells that build functions. You probably have local functions, and then different local functions build the full heart function. So do you need to split off the concept of population at the organ level and then at the individual level, meaning that first you need to capture a population just for one organ or for a specific part of the organ, and then try to focus on inter-individual variability? How do you conceptualize this?
Yeah, because in a way it's really, first of all, I guess the question is again, what is the purpose of building this? And of course, one is that if you would like to make a model of the heart, your own heart, for example, of course, then you have to kind of figure out what is the kind of basic kind of functionality of your cells. But then our heart has different kind of cells. Of course, they're atrial and ventricle and some other types of cells, but they are also different. And of course, one can make a population of cells that constitute single person heart. And if you think then about variability of healthy population, then we can kind of extend that. And if you think you have certain kind of population that has some genotype, then you can try to make population of that. So I think Blanca Rodriguez from Oxford, we are also collaborating. She has done quite a lot of work on this kind of personal heart level as well, building cardiomyocytes and populating a heart. So if you're interested in it, there's some papers to that as well. But definitely you have to also know what for you are building. Are you building a single person kind of variability population? Are you building it for a certain genotype? Or if you look for certain disease that are on phenotype? But
yeah,
because it's an important question, really.
Yeah, thanks. Because according to your experience, so you've shown, for example, so the action potential, so of a cardiomyocyte, normal one, and a diseased one. And basically the upper peak was larger for the unhealthy cells. So now, if you put that in a context of cell collection, would you have just a natural variability, a noise that makes this difference of peak irrelevant, or on the contrary, would you have added contribution that makes the difference even larger. So do you know that or not?
Trying to kind of grasp the... of course, there's all kind of problem is the data as well, that whether the kind of observation in vitro or observation in human level is for chance of this person, or is it if you have 10 patients or 10 cells or 100 cells, what is the really representation of that population that you're using as a reference? I think that is the first question. Like, but when we are building these population models, of course, we use the data from literature, and they might be from 10 labs, have done the work and look the variability of the. I think there may be some like 20 different parameters at least, from calcium and from voltages, that use for building these. But still, it's kind of, we can guess that might be enough. But of course, whether that data that you're referring your simulation is enough for really having like a real human population level prediction. Of course, we don't know yet, because in a way, if you have population study on human population, you might have 1,000, 10,000 people or 100 people. And it's the same kind of thing when you have in-silico population, or you take certain population study for living persons. It's the same about how representative your data is to the real human population level, or are you targeting a certain single person with certain genotype. Did I answer your question? I'm not sure.
No, you did.
Okay, thanks.
You did, definitively. Yes.
And have you had studies really getting cells from different type of people, like, gender, just race, and I mean, do you see interesting difference at the cell level? And
yeah, there's, of course, the kind of availability of these iPS cells is still a kind of question, and I think we have like 200 persons data bank or cell bank, mainly from different cardiac diseases. So they are male and female, and that is one important question that people are now starting to actually answer, looking at these cells, but it's not being clarified yet. So there's definitely a question about, for example, gender issue, especially because the many diseases, especially like in cardiac disease, the gender is a huge effect on how the disease is. What is the phenotype? You can have male and female, same genotype, but how the cardiac disease is observed in the person is totally different. And there's a lot of work that hopefully this in vitro, in silico kind of way can kind of help as well.
And just, I think, to finish, but I mean, I was having a question. Okay, how do you see this field in 10 years? In fact, in your slides, it was in 3 years, 2025.
Yeah, no.
I would ask you to be a bit farther than that. I mean, because you were showing initial results really like a bit more than 10 years, and I guess that you wouldn't expect what you have arrived now.
Yeah. I guess what has happened, as I said lately, is that the field of research has been kind of exploding. So I think that there will be more innovations as well in vitro, but hopefully also this how to use in silico incorporation this in vitro devices, and not just cardiomyocytes in many areas. I would answer this maybe after a week when after seeing the World Summit on in vitro world next week. But definitely, I think it will have a more rapid kind of progress than last 10 years.
Mr. Chairman, am I allowed?
What? Yeah.
No. Yeah, that was about the genotype. You know, that's. I don't remember for which cardiac disease, but I think sudden death, some people have identified gene panels. So that's similarly like cancer, so that they can assess your risk of. So would in a really multi-scale or heterogeneous modeling perspective where you couple these single cell models to bioinformatics, would actually then the knowledge of this gene panel help to be more predictive? Yeah,
especially, of course, it's really that your model should have certain kind of functionalities that can be kind of linked to certain genes. And if that can be done, of course, now we know a lot of genes that are related to, for example, ion channel mutation or something else those are easy. But having statistics as well, that what could be the predictions here, why these persons maybe there can be linked to these certain genes. I think that's really something important that we should really combine, because now I think the world has been a little bit separated, like what is happening in vitro in this kind of silico world and in bioinformatics. And that definitely is an area that we should look much more in the future to explain things and look into kind of pinpointing the population that are in danger, for example.
Perfect. So, well, let's thank Professor Hyttinen for the wonderful lecture. Thank you.
Thank you.
Okay. Okay. Well, now is the time to introduce the last speaker of the day. And for me, it's an honor to introduce you Jack Hoyhott, who is the chair in the Bernal Institute of the Biomedical Engineering program at the University of Limerick in Ireland. His research is focused on poromechanics of swelling materials. So for all people that is working in IVD modeling and cartilage modeling, so as a matter of fact, all our research is based on his contribute, so we are really happy to have you here. And also he is a founder of the International Society of Porous Media that actually focus on the use of all these poromechanics, let's say, tools, not only in the biofield, also in different areas on the industry and engineering. So welcome, Jap, and thank you very much for being here, and the audience is all yours.
Thank you very much for the nice introduction. It's a pleasure for me to be present here on this convention and this interesting number of people studying the numerical applications of numerical methods in clinical applications and biomedical applications. What I am going to do is a little bit different maybe than what some other people have been presenting here, is that I will go into how to improve the tools that we use in biomedical engineering for numerical simulation so that we can increase the reliability of these tools. To give a little bit of an idea where I am, I have a secondary position in Holland, but my main position is in Ireland and is part of the Bernal Institute. And the Bernal Institute is an institute mostly privately funded from donations and from industry, focusing... Oh, my thing is gone. What happens? Was a problem with Microsoft PowerPoint. That's not good. What's happening now? PowerPoint.
C'est ton PowerPoint qui est sorti?
Disappeared. No, not new present. Open recent. Barcelona. You have it coming back? Si no.
Est-ce que sinon no te vas a presentar?
Est-ce que s'havia tancat el PowerPoint?
Però de tota manera ho ha de deixar en mirror, perquè ho té com a segona pantalla. Igual seria més fàcil. Bueno, però aquí té les notes. On va le mettre un peu plus haut.
Let's see if it remains stable. So Bernal Institute is a project of about 100 million euro, which aims at employing about 400 researchers, mostly funded from donations and from industry, but also a little bit from the government. And there are about 6 or 7 chairs there, one of them being biomedical engineering. And a significant part of the research relates to pharmaceutical science, but is in fact much broader than this, including material science, molecular biology, and energy technology. Now, as far as what I am doing in the Institute, I'm focusing basically on 2 areas. One is intervertebral disc research, and the other one is cardiac mechanics. And both of them are focused on having numerical simulation tools in relationship to experimental setups in the lab. And for the intervertebral disc, we cooperate a lot with Christine Lemaitre from Sheffield and Theo Smith from University of Amsterdam. The themes that we are researching involves voltage-gated ion channels, piezoelectricity, scoliosis, and finite element analysis of electromechanical coupling in the disc. Apart from that, we have a significant line of research in cardiac mechanics, in which we try to fill a serious gap into cardiac mechanics in the sense that we are including coronary perfusion and trying to make a tool which can be used, maybe not for patient-specific problems, but at least to evaluation of cardiac devices. Now, if we look at what happens when we start on finite element analysis of specific organs, such as the intervertebral disc, my experience, and usually that is not very much reported in the literature, my experience is that quite often, because of the complexity of what we are studying, including the fact of heterogeneity of the domain, the anisotropy, very large deformations, discontinuities like contact or fracture, quite often people using standard tools such as abacus or ANSYS or whatever, they get convergence issues. In fact, when we talk with the abacus people of what we are doing, they think, oh, that's never going to work. So obviously, there are publications, nice publications showing, oh yes, 3D analysis, and we took ligaments and contact and fracture, etc. And all of this seems to work. But the reality is that the student has been trying and trying and trying and finally found something which works and then publishes it. And doesn't mention that he checks so many times to actually find something which actually converges. So I think if we want to move towards clinical application of finite element analysis, it should be absolutely reliable and stable. And even in very challenging areas, such as large deformations, strong heterogeneities where the material properties changes orders of magnitude, such as between bone and cartilage. And all of these should not be an obstacle to actually have it running. And so I'm going to talk about what we've been trying to do to improve the situation as it is today. And in trying to do that, we realize that developing finite elements rather than using existing finite elements, when we try to develop finite elements, we have to think across disciplines. We cannot afford to say, oh, we are going to develop specifically for intervertebral disc degeneration a special software which is completely geared to that particular application. It doesn't work because you don't get funding for this. And immediately they expect you clinical applications, some development of a device or something, and you can't get it funded only on that application. But the reality is that when you think of finite element development, this can have a very broad application. And you'll see that I will discuss not only disc, but also application to gels, and gels in particularly in relationship to diapers, nappies, and also in terms of geomechanics, there is the equivalent of herniation of a disc, but then known in geomechanics as hydraulic fracturing. All of these different fields, they need to talk with one another to create the finite elements which are reliable and can be used systematically without worrying about the convergence of the equations. So I'm going to touch on this. I'm going to touch on fracture as well. And I'm talking a little bit about perfusion modeling, where we use similar methods which we are used to have in swelling media. So if we think about swelling media, you can draw many applications, many different applications in terms of the stiffness of the material and in terms of its swelling propensity. And then you see, for instance, sandstone is something very stiff and not swelling at all. If you go down to gels, then you have PVA, which is somewhat swelling, but not so much. HEMA may be a little bit more, and you may have ionized gels, which are much more up in the corner there, but cartilage and disc are somewhat swelling and in the order of megapascal stiffness. But if you talk about intracellular space, you're up to kilopascal stiffness and very high swelling propensity. And especially in that corner, upper right corner there, we get really into trouble. Why? Because the high swelling and high deformability makes the models and the materials extremely versatile and even sometimes unstable. Now, in this area, it's not a small area because most of biology happens intracellularly in cytoskeletons and nuclei, cytoplasm. In those areas, you're down to kilopascal stiffness. And then what you find is that abacus doesn't work. That's the issue. So what to do? And actually 90% of biology takes place intracellularly. So getting funding for intracellular modeling of poromechanics, forget about it. What you can do is use applications from the industry. And now a very famous application, which we all know, is diapers. Diapers are highly swelling. A very efficient product, which all of you who have had children had used a lot. And if you go down to what happens inside the diaper, you find small beads of hydrogel, which swell about 30% of their volume, 30 times their own volume. And they're actually surrounded by some kind of a cross-linked layer on the outside, and you see as it is swelling, and this is actually a bead of this type inside the diaper, when it is faced with a gush of urine, it starts swelling, but before swelling, it has to get this surface shell to crack. And it cracks into pieces, and you see it, oh no. What happened now? It doesn't seem to like it. Recent, we start again. So that's another example. Oh, we can't see it. Wait a minute. I go in presentation mode. That's the issue to get it done. Now, this is also a small bead of such a diaper, and the small bead is looked upon by a very simple method. You know, many optical methods, you use very advanced optics, etc. Here we did nothing of very advanced optics. We took the camera of a mobile phone, you know. You take off even the lens, you do away from the lens, do away with the lens, and you put the bead straight on the chip. On the chip, the CMOS chips, which observes the photos that you take every day with your mobile phone, and you put it on there, and then you look directly on the particle as it is lying on the chip. And so, I hope this is going to work. That's it? Okay. It's almost running away, but you can see very clearly that it has what is called the cauliflower effect. So it swells one side and then the other side, and so it's a very non-homogeneous swelling of the phenomenon. So you see surface instabilities coming up, and to illustrate this even a little bit better, I can show you another video which we've been making of a gel of a cylindrical form, and we move it from high salt concentration to low salt concentration, and then it starts swelling. And it starts swelling, but it develops surface instability, which here you can see very clearly because it starts with a cylinder. And what happens, in fact, is that the outside swells first and the inside doesn't, because the water cannot reach inside. And then the kernel and the outer surface needs to remain in contact with one another. And the way it does, it goes into surface instability and later into a kind of saddle shape. So it is convex to upwards in this plane, and in the plane perpendicular to the picture, it is convex downwards. And so we have a kind of saddle shape, and progressively it is swelling. In the meanwhile, the ions go out of the gel and the water goes inside, and they compete actually with one another. And at some point it goes also into the center of our gel, and progressively then it goes back into the original cylindrical shape. And going back to the cylindrical shape demonstrates that our gel is purely homogeneous. So we have a purely homogeneous gel, but still it goes into those mad shapes. And one of the challenges that we took, and particularly in perspective of what is happening inside the diaper, we actually wanted to be able to simulate this. And I must agree that still now we didn't succeed in simulating what is happening here. But we got some way. And now, because of the special relevance to biology, considering that 90% of biology happens intracellularly, and we pay attention to this also from the point of view of biological application. Now, if we look back in time, you know, I'm working at the Bernal Institute, and Bernal was actually the student from a Nobel Prize winner and also the teacher of 2 Nobel Prize winners. And he worked on proteins. He was the one person in the world who has been the first to apply crystallography to proteins. And one of the most important things that he concluded is that if you want to analyze the structure of a protein, you need to study it in water, because interaction of protein with water is absolutely essential for the structure of the protein themselves, and to understanding the protein themselves. So what we use, basically, practically speaking, in order to study these gels and intervertebral disc is theory of finite deformation of porous solids. And it's actually a very old theory because it dates back to 1972. And on top of that, we use the insights from Fleury, also some Nobel Prize winner on swelling, which who argued that you need 3 free energies. One is elastic free energy, the second is mixing free energy, and 3 is the ionic free energy. Ionic free energy is the one which was responsible for Donon osmosis. Many of people working in cartilage and disc know they work with Donon osmosis. So this free energy of ions is very, very important. Mixing energy is in disc and cartilage is known under the name of exclusion phenomenon, and is about responsible for 10% of the swelling in cartilage and disc. Elastic free energy is just the LCST, so that's quite obvious. Now, in terms of gels, Mixing becomes very important when the gel is dry, and increasingly the ionic part may become important if it is an ionic gel, and as swelling goes on, the ionic part becomes more and more important. So we have basically a double layer around every protein or every polymer, and negative charges with compensating positive charges in the fluid, and this is basically what generates most of the ionic swelling, namely around the positive charges there are a lot of water molecules around it. And we can talk about the bi length, namely the thickness of that double layer, which becomes more as this outer salt concentration becomes less, and less if the outer salt concentration becomes more. Now in gels, unlike disc, you can say that ion diffusion coefficient is typically much larger than pressure diffusion coefficient. Why? Because pressure diffusion coefficient is linked to the stiffness, and because the stiffness is so low, you get low pressure diffusion coefficient, much lower than the ion diffusion coefficient. This relationship does not hold for cartilage and disc. So when we can say this, the nice thing about it is that we can say that the ions go immediately in equilibrium, and we can forget about all of ion diffusion altogether, and we can only focus on this pressure diffusion, what we exactly going to do, and this leaves us with only a fluid and a solid. And then there are equations, and for those people who are at home with equations, this is momentum balance, this is mass balance, and this is Darcy equation. Flow times permeability is equals to permeability times gradient of chemical potential. And we can write these equations down, and we can try to solve them. And the swelling we account for by means of an ionic component, a mixing component, and an elastic component. And I want to go into the details of these equations. But what you do, if you put it into Abacus or any other standard software, you see that everything goes fine as long as you're around megapascal. Megapascal, it's okay, you manage because it converges. But as you go down, and typically this happens inside cells, is that 55 kilopascal, you have most are already failed, and 15 kilopascal, you get no answer at all. So you get really into trouble down to that level, and not only you get into trouble when you go to low stiffnesses, but you get also trouble when you go into heterogeneous materials. You get into trouble when you have strong anisotropy. Many issues are created as soon as you get out of the standard range of application. Now, the question is, why is that so? And can we do something about it? Now, the trouble is that you cannot do something about it inside Abacus or inside ANSYS or whatever package, because you have to be able to look inside. And so what we did, we first analyzed very carefully why the problem comes up. And what we found actually is that one of the key issues here is what is called local violation of mass balance. Local violation of mass balance. Just a very simple experiment. Many of you who have been working on cartilage will know, is consolidation experiment. Consolidation, confined compression. We have a sample of whatever cartilage or whatever lying on a filter. We put a piston on it and we push out water. And as we push out water, we get a settling of the cartilage because water is pushed into the filter. And we can also try to simulate this. Then we have a finite element mesh, a few elements one on top of the other, and we define pressure. And typically we would assume pressure to be linear inside an element. So we have piecewise linear variation of pressure, and then we derive our flow with Darcy's equation. Flux equals gradient in pressure times permeability. and then we get a flow for one element, another flow for second element, another flow for third element. Now you see immediately what is the problem is that this element pushes out so much fluid, according to that red line, into the next element, and the next element only less comes in. And same thing with a jump here across the elements, you have a discontinuity. So the flux out of one element is not equal to the next one. And this is not an issue. As long as it is globally accounted for, most of finite elements you can use abacus without any trouble. But when you go to very large deformations, you get really into trouble. And so what we do is basically using a trick which is not new at all. It has been used for years in fluid mechanics. It's the use of what is called Ravier-Thomas elements. Ravier-Thomas elements described in very difficult mathematics books on finite element analysis. And the simplest one which you can think of, which accounts for local mass balance, forces a local mass balance to be true, is one of this type. You have, in 2D, you simply have a square element with on the corners unknown displacement, and you account for the flux through each of the sides, edges, as degrees of freedom on top of the displacement. So where here we are using pressure, here we are using the fluxes across the edges. And we are going to use that element because this one, we'll be able to force the fluxes from one element into the other element to be same, exactly same. And that's what we do. And we force these two fluxes to be exactly same. And the way we proceed is as follows. If you take now the example of a triangular element, you have the displacements unknown in the corners and three fluxes. And on the top of that, we have one pressure, or in case of swelling, it is a chemical potential in the middle. And we can write down the full equations as we have them from the equations I just showed you. Conservation of momentum, conservation of mass, and Darcy's law. And we can solve those equations, but as you see, we have more degrees of freedom than if we had taken pressure simply, because we have those fluxes. And what we do now is enforce these fluxes coming out of one element to be exactly equal to the flux into the other element. So we have two elements next to each other in a big mesh, and so we are forcing this Q3 to be equal to Q3 star by being of a Lagrange multiplier. And what we find is that we can add this condition to our set of equations. And then it becomes a really big equation set with Lagrange multipliers and chemical potential and flux and displacement. And this one will be able to enforce local mass balance. But now we do a further trick, and that's what is called hybridization, where you eliminate the mu and the delta element by element, the Q and the mu element by element, and what you're left with is displacement and these Lagrange multipliers. Now, what are the Lagrange multipliers? They are along that those edges. They are basically the pressure in normal porosity or chemical potential if you include the swelling. So we use this formulation, and we are going to look at a small piece of gel and allow it to swell. And you see, this is what happens. We have tetrahedral elements in 3D, and it can swell to multiple times its own volume, and we get very stable results. And other example, which is maybe even more illustrative, it is a ring fixed to a fixed surface, brought to swell, and it goes into surface instability. That's basically what I showed you experimentally. You can go into significant surface instability and very large deformations because of this creasing going on. And I can assure you, you will never get this kind of calculation done with Abacus. But it is typically what happens if you are down to very low stiffnesses, you get surface instabilities. And this kind of analysis is the one which is happening. And you can actually verify also experimentally that the phenomena that you calculate correspond to what you measure. You can plot one relative to the other, the swelling propensity and the stiffness, and then you get one side of the curve where there is surface instability and the other side where there is no surface instability. And same thing with numerical result, stiffness versus swelling, and one side is unstable and the other one is not. So, and now we can go back to our simulation, look at what happens when we take down the stiffness down to 15 kilopascal, and you see that most of our simulations nicely converge. And so this is what we want. We want an absolutely reliable finite element analysis which can be used routinely without each time getting responses, oh, this calculation doesn't work. So going back to the cylinder that I showed you in the beginning of the presentation, we can show that as we take it to lower salt concentration, we get swelling, but also surface instability. You see very clearly the surface instability. It's not at all in the order of magnitude that we had experimentally, but still there is surface instability, and we are working on getting this even more pronounced at what it is here now. So what happens during that swelling is, in fact, underneath the surface, you have a strong compression building up. Compression, here you have surface effective stress. It goes strongly into the negative before rising up again. And this strongly going into the negative is the source of the surface instability that takes place. Now, things are kind of complicated also in terms of material properties. Material properties, we've done experiments, detailed experiments on this, and MIT has also done experiments on this in cartilage, is that if you disallow cartilage to swell and change the salt concentration within which it resides, you will find that the cartilage is going to stiffen as the salt concentration goes down. So we usually think of, material properties, oh, they depend on strain. Stiffness is more on higher strain than lower strain. Now, even if you keep the salt concentration, and you keep the deformation absolutely constant, but this time you change the outside salt concentration, then you change basically the thickness of those double layers. So this is the Dubai thickness, here in high salt concentration, here low salt concentration. It changes the thickness of the double layer, and part of the stiffness of a gel is related to the thickness of those double layers, and not just to strain. Even if you constrain the gel and disallow it to swell, you get a change in stiffness. And we did actually experiments. Here, this is a gel enclosed from all sides within a torsional testing bench, and enclosed within a ring, so that it cannot swell or shrink, but we can change the salt concentration in the bath. And then you find changing stiffness depending on the salt concentration. These are 3 different salt concentrations, 0.03 molar, 0.1 molar, 1 molar. As you change the salt concentration, even at constant strain, you still get a different stiffness. And this kind of result has been found by us for gels, but by Alan Grodzinski in MIT for cartilage. Here we have a dependence of stiffness on salt concentration as such. And you see here shear modulus plotted versus ionic strength, and you see quite significant changes in stiffness. all of these, we can plot, build this into the model so as to represent really what is happening in such a gel. And automatically, when you assume these shear modulus to depend on salt concentration, also electric potential depends on strain, which may be extremely relevant to mechanotransduction as well. Now, a second theme I would like to talk to you about is fracture. And fracture is a phenomenon which is extremely relevant to disc degeneration, because most of disc degeneration is associated with fracture. And fracture means that very locally at crack tips, there are very large strains, even in relatively stiffer environments such as disc or cartilage. And the strains are large, and there also we expect local mass balance violation. Now, the disc is known to be cracking or tearing as it degenerates, and usually this tearing is not very much associated with loading. It's actually intrinsically associated with aging, swelling, loss of fixed charge density because of the degeneration. And so this fracture is, on one hand, relevant to biology. On the other hand, finding also funding for this kind of simulation on fracture using coupled to swelling is a difficult one. Now, I showed you in the beginning the gel from the diapers, and you see also there was fracture. So we got actually funding through Procter & Gamble to do this kind of simulation, and we started on fracture propagation in swelling media. And I will start with an experiment. An experiment relates to a gel, and the nice thing about gel, you can look inside it, which almost you never can do in a solid, but here you can look inside it, and we have the gel, a piece of gel with a small crack initiated in it, fixed to both sides to a drawing bench, the grips of both sides of the drawing bench, and you're going to move apart one side compared to the other and observe the cracking inside the hydrogel as it propagates. And so this is the gel. You see the initiating crack here, and we're moving apart the jaws, and you see that it progressively propagates. And we hope it to be a one-dimensional phenomenon, but you see how it is a very complex thing happening at the crack tip, and you should think that similar things happening in the disc and to cartilage. But if you look very carefully, you will see that it doesn't propagate in a continuous fashion, but much more in a stepwise fashion. Suddenly it propagates, then stops, it propagates, it stops. And we find this even for just strictly homogeneous material. So gel is homogeneous. There is nothing like inhomogeneity. And if you look at the surface, you find that there are actually teeth along it, which also indicates that it is stepwise progression. And so we also could show that the distance between the teeth depend on the Young's modulus. You did many of these experiments, and you see that Young's modulus, as Young's modulus increases, you get a longer distance that it propagates each time between the steps. And the time it pauses does the opposite. It is higher for low Young's modulus and lower for high Young's modulus. So interesting finding, but we tried it also to simulate this. And you say, well, we use XFEM. XFEM is an advanced method of finite elements, well known to do fracture propagation. And people in concrete and in steel, etc., they use it. But we wanted to use it for poromechanics. And so this is a crack inside a poromechanical medium. And the XFEM, nice thing about XFEM, you don't need to do remeshing. You just do enriching of nodes. as you propagate through the mesh, you actually progressively enrich nodes as you go through the finite element analysis. And so you get at some point a whole path followed by the finite element method and progressive enrichment of all the nodes which are tainted in black here. And what you basically do is using the same thing as regular finite elements, using displacement degrees of freedom and pressure degrees of freedom, but you enrich the displacement and you enrich the pressure by means of an extra discontinuous shape function. And this is very standard stuff, only what we did along the crack, we had pressure defined inside the crack, but also a pressure defined on the left side of the crack and on the right side of the crack. We had 3 pressures in each point, because the thing is that we expect these 3 pressures to be systematically different inside gels. And so we allow fluid to flow from the crack into the formation, into the gel, and from within the gel, it is flowing as well because it's porous media, and also within the crack it is flowing. So these 3 things are accounted for, and also there we got funding from again another source, and hydraulic fracturing is a very important thing in oil industry, and so also there we use fracture. And this is a simulation of a fracture propagating inside a porous medium. And you can actually show that under gravity, this fracture propagates, but typically it's going to propagate horizontally, even if you start vertically. And I can show you more simulations. This is a small initiated crack, initial crack. We introduce some fluid pressure inside, and then it propagates progressively, goes into existing cracks which are already into the structure, and then you get exchange of fluid between the new crack and the existing crack, and so progressively you get propagation of fracture. And also here we tried 3D, and we can show that 3D also something is possible, but also there the stability may be disappointing. Now, we go back to our experiment. Our experiment is a hydrogel with an initiated crack, and we simulate in finite deformation this time the propagation of the crack inside the gel, and we can plot displacement or pressure, and we have a propagation of a crack which is definitely, as you can see, in the range of large deformations. And what we find is spontaneously that our system predicts stepwise propagation. It predicts stepwise propagation. It does not propagate continuously, but stepwise, despite the fact that the material is strictly homogeneous. And we find also that propagation length between the different teeth is increasing with shear modulus, which is consistent with experiment, and also the pause time is decreasing with increasing stiffness. So also there we find consistency, at least qualitatively, what's happening inside the gel. Now the question, why is it staccato? What happens is in poromechanics things are kind of more complicated. In a solid, you have crack. Ahead of the crack, what happens is that there is the material is somehow torn apart. It needs to be torn apart because that is going to allow the crack to propagate. So you get a triaxial tensile state ahead of the crack. And this happens in poromechanics also, only it is the fluid which is under pressure. And this under pressure attracts fluid, and the fluid goes towards that point, and then progressively the fluid pressure goes up again. And what happens is that this tensile load is transferred to the solid, and then you get propagation. And again, you have the same thing happening again. first the fluid taking up the tensile stress, and then progressively handing it over to the solid. And it's in this way, we think that we can understand what is happening inside this gel as it is propagating. So we have fluid flow towards this center, and progressively we see that the effective stress is increasing. Now, what you can show in gels is that typically the propagation distance square divided by the pause time is in the order of magnitude of the pressure diffusion coefficient. What is the pressure diffusion coefficient? It is the hydraulic permeability times the stiffness. And you find, therefore, that you can assume that this stepwise propagation is directly associated with fluid flow inside our gel. Now we can do also simulation of several cracks at the same time in finite deformation, and you see them propagating and going inside, as you would expect it to happen in a gel, but also as you would expect it to happen in an intervertebral disc. And we can actually show also that without loading from outside, a gel can start cracking. And this is what happens in the disc. In the disc, you get crack propagation, and you can show that different people with having very heavy loaded spines and other which have low loaded spine, whatever happens, it's always so that cracks develop inside the disc. And this shows actually a simple experiment. This is a cylinder of a gel, but which has been cross-linked, so it cannot stand swelling very well, so it goes in a crack. And this is actually in another view of the same sample, but produced by a mirror. So here you get the swelling, and what happens, it cracks into pieces. So the opposite you can do as well. You can actually deswell the gel, and it goes into pieces as well because it cannot stand this kind of cracks. So this is kind of idea that we can create to understand what is hydraulic, what is disc degeneration with time, using this phenomenon. But also there we are using, on one hand, the intervertebral disc application, on the other hand also oil field applications, where also there it is known that shales, which are also swelling, can actually cause a lot of trouble to oil companies as they are drilling into the soil. Now, a completely different applications, which we are seriously working on, and also using mixed hybrid finite elements, using Ravier-Thomas elements, is a application in the science of in the world of cardiology. In the world of cardiology, there is a lot to do about simulation in 3D of the electrophysiology and the contraction of the heart. But if you think very carefully, damage of heart, failure of heart, is in 95% of the cases associated with fluid flow. Fluid flow, coronary flow inside the heart being disrupted for some reason, and somehow the heart, the blood flow cannot reach certain areas of the heart. And so we want actually to integrate this complex flow phenomenon inside the heart using standard methods from poromechanics in order to simulate this during contraction. This is here the system of coronary flow, with the left descending coronary artery, the circumflex coronary artery, and the right coronary artery, all feeding the heart from the aorta, which you see here on top. Now, What we can do is, Eindhoven has a very advanced model to simulate in 3D contraction of the heart coupled to electrophysiology. And we take that model, and we have also an axisymmetric model of a heart, left ventricle. And we take that model and we add to it a coronary circulation model in which we distinguish between arteries, arterioles, capillaries, venules, and veins. And we simulate blood pressure, blood flow during contraction. This is during end diastole and ejection. We are working on having simulation of blood perfusion inside a beating heart, so that we can do prediction as far as the effect of specific cardiac devices on this phenomenon. Here, this is a view of different compartments inside the left ventricle: arteries, arterioles, capillaries, venules, veins, and we can simulate this as a function of time. This is done specifically here for end diastole, but we can take different times during contraction to simulate this coronary blood pressure and coronary flow in order to predict failure of the heart. This is ongoing work, and as a conclusion, I would like to tell you that mixed hybrid finite elements has added value compared to normal UP formulation as available in Abacus, for poromechanics, particularly for very large deformations, but also for non-homogeneous media. At low stiffness and high swelling propensity, gels exhibit surface instability. Shear modulus of gels depend both on strain and ionic concentration. There is evidence of staccato crack propagation in homogeneous gels, and the XFAM method that we developed is able to predict the staccato for propagation of gels of cracks in gels. Multiple cracks, merging of cracks, bifurcation of cracks are within reach of XFAM simulation, and coronary circulation modeling in contracting heart may lead to virtual environment which allows us to simulate cardiac devices before launching into animal experiments and clinical trials. So I have to acknowledge many people who have contributed to this work, and also many companies who had helped in financing all of this. So maybe there are many questions.
Wow. Wow. Thank you very much, Jacques, for this amazing talk today. So we have time for questions. I hope that.
Okay.
Thank you very much for the talk. I would like to ask about this change in the same shape functions that you mentioned. Do you also apply it in the displacement degrees of freedom or only in the fluid flux or pressure?
You should think, if you think for solids, you write down classical equations of solid deformation using displacement as degree of freedom, automatically complies with local mass balance. Why? Because you make a mesh and there are no gaps between the elements. The elements stick to each other very nicely, even if they continue they continue deforming, they stick onto each other. So the mass balance is automatically accounted for a solid. What is not accounted for perfectly is the momentum balance. Momentum balance is accounted for on the average. So there you are not perfect. For the mass balance, you are perfect in a solid. And when you add fluids, you do solid fluid simulation, you comply with mass balance for the solids perfectly, and your solid, your momentum balance is going to be accounted for on the average. But for the fluid, you did the opposite. because your pressure is continuous. There are no gaps in the pressure. It's not that one side of an element has another pressure than the other side. But you have problems with mass balance. And the reason for doing this thing of Ravia-Thomas element is to account for mass balance locally for the fluid. And the reason why we get better results is because we account for this mass balance. And it's an old trick which is used in standard way since many years in fluid mechanics. So all the computational fluid mechanics packages account for that. But in poromechanics, people never minded about that. And so when you account for that, you have a much better model, a much more stable model. And the computational time is same. It's not that we are doing something much more complex, because we just have the displacement and then some, here, Lagrange multipliers, but it's basically pressure or chemical potential. It's same thing. In size, it's not more complex, only it's a little bit more complex in programming. And we've been pushing the Abacus to build it into their standard package. They promised us to do so, but I haven't seen anything happening yet. Usually you should think if you give them the equations, you know, they solve it in 2 days' time. One employee from them can have it inside Abacus. So it's not that it's a lot of work, because it's extremely routine in putting new elements into Abacus. But you know, it's business, and as long as it's not clear to them they really need it, then it doesn't happen. Now there is one thing, you know, developing finite elements, the thing you need is to have companies behind you, because we are customers of Abacus, but they give us Abacus almost for free, because we are educational and etc. And we don't have money, etc. So we get it almost for free. Now, if you go to Procter & Gamble or Shell or whatever kind of big companies, they pay, I think, maybe 400,000, 500,000 euro per year for their subscription to Abacus. And what they have people walking around in their research labs paid by Abacus. And they're just helping people. So what you need in order to get this kind of developments into commercial packages is have the support from these big customers. Because Abacus worries about having Procter & Gamble as a customer, but they don't mind about University of Eindhoven, or it's of no value to them. So you need to have support from them. And one of the issues that we have when we really move towards biological application is that we need to have support from a multinational to support this. This whole thing of blood perfusion, getting into a commercial package is one thing, but the other thing is getting it supported by the company. And you get it supported by the company only when there is a significant market for them. And you have to justify that this market is big enough. Now, Abacus has started a project on living heart. And what happened is that one of the guys from Abacus, he was manager there, and he got a daughter. And this daughter had a congenital heart disease. And then he was watching on the pediatric cardiologist studying the MRI of his daughter and looking what he's going to do. And he was flabbergasted that this guy was going to cut into the body of his small baby daughter without doing any computation whatsoever. He just started cutting. Now, if you think about what we do, I came here by flight. Now, this plane, with which I came here and which many others used, we are pretty sure that when we leave from Holland, we're going to arrive in Barcelona without much trouble. Why? Because every small part of a plane is calculated by means of finite elements, on fracture, on failure. Every small piece is calculated everywhere. And therefore, we know for pretty sure that we are going to arrive safely into Barcelona. Now, it is incredible that when you start working on a heart, that you have no computational tool whatsoever. One works on feeling, starts cutting and reconnecting and things, no computation whatsoever. For failure of heart, there is no computational tool, nothing. Because it's disruption of blood perfusion, and nobody does blood perfusion in finite elements. People are used to do stresses and strains. So people are doing stresses and strains because they're used to stresses and strains. But a cardiologist doesn't know about stress and strain. He wouldn't even know the difference between stress and strain. So while he knows everything about blood perfusion, and the finite element people don't know about blood perfusion. I talked about blood perfusion with the living heart people from Abacus. They never heard about it. They don't realize it exists. They just do stresses and strain in the heart. So there is really a problem of communication between, on one hand, cardiology, and the other hand, the engineers. And they talk simply a different language. They do different things. And that's why there is a real serious issue, while the reality is that there are 18 million deaths a year on cardiovascular disease. So there is a lot to do about it, much more than those planes, because those planes, you know, there are many planes in the air, but there may be 1,000 deaths a year, a few planes falling down every year, something like that. Now you can say that's very bad, okay. But 18 million is much worse. So there is an urgent need for finite element analysis and comparison with experiments, prediction of what is going to happen, and it's very difficult to get it going. It's really a challenge. And so we are working on it. We think that it's possible to do it. And I really believe that if we continue working on it, we're going to convince the people. But how many years it will take, I don't know.
Thank you.
I think just a comment, that's just amazing what you've said about this blood perfusion stuff. A few weeks ago, I was having a beer with a friend of mine. She's a vascular surgeon at the hospital clinic here in Barcelona.
Yes.
And she said exactly the same thing. Say, what we are lacking are model computations for blood perfusion in order to improve our interventions. So I was really basically struck by what you've said.
Yes, yes.
Such convergence.
Yes.
Terence.
Thanks very much for that. Just along the same lines then of blood perfusion, but going back to the disc, and I'm just wondering if you have any thoughts in relation to modic changes and the subchondral bone and the effects of the hypertension in that area and the pressure of blood perfusion in that area, and whether that can have any link or mechanical effects in terms of vertebral end plate disruption or end plate defects.
I think if you think of pressure in the heart and in arteries, you're thinking about a few kilopascal. Now, if you think of pressure inside the disc, you're talking about much higher pressure. You're thinking of many megapascal. So the order of magnitude is much different. So if you think of what is happening at the end plate, the osmotic pre-stressing and the loading on the disc is probably much more important than the pressure into the arteries. Now, what definitely can play a role is what is happening on the level of capillaries. And I think there are also huge gaps on what is exactly happening on the level of the microcirculation. I realize that microcirculation just physiologically is very poorly understood. And it is extremely important, not only in disc degeneration, but also for diabetes. Many issues with cardiac function and also brain is associated with microcirculation, and usually neglected for the simple reason that if you look at X-rays, you see large arteries. You can see stenosis, occlusion. Immediately you can say, well, I'm going to put a stent and problem is solved. Now the reality is that a large part of the patients don't have any stenosis or occlusion and still have problems with heart or brain function. And you cannot find any lesion, and especially ladies who complain about heart problems, they don't have lesions, not observable lesions. But on the level of microcirculation, a lot is happening, which we never see. And the cardiologists and the spine surgeons and the brain surgeons don't see what is actually happening. And there is a huge gap, I think. Even simply of physiologically understanding what is happening. One of the things we are researching now is, and that we do with microfluidics, we are trying to understand the following phenomenon. It is known that there is something like Farus-Lindquist effect, namely that the red blood cells are always flowing a little bit faster than the plasma. It's a little bit faster, maybe 10, 20, 30, 40%. And the difference becomes more as you go to smaller blood vessels. Now, in capillaries, you find that during exercise, the flow in the capillaries is for red blood cells, maybe 30, 40% faster than plasma. But if you look at a resting muscle, for some mysterious reason, suddenly this ratio becomes completely different. The red blood cells are going 3 to 4 times, 5 times faster than the plasma. And nobody understands why. This is not just convection. It's just something special happening there. And we are trying to illustrate this on microfluidics. But this is just a very simple phenomenon which is observed everywhere in microcirculation, and it's not understood. So there are things which need to be researched on that level, and I think there is a very great need from the clinical point of view, because cardiologists don't know what to do. And I think for disc, it may be a similar issue. What's happening exactly at the interface between the cartilaginous end plate and the vertebra is, I think, extremely important. That's one thing, and probably poorly understood, because we don't know what's exactly happening. So
yeah, thanks very much.
Yes. Yeah.
Yes. I have, well, thank you for the talk. I have a question related to the fracture simulations. Do you think that could be possible to include in these fracture simulations hydrogels containing cells and kind of predict how these fractures can happen with cells inside the hydrogels?
Oh, putting cells inside the hydrogels.
As a 3D culture model, for example.
Yes. I think it would be worthwhile doing, because I would be pretty confident that you can measure that cells sense the fracture. If you would kind of doing something with immunohistochemistry or whatever, showing what is happening with cells directly at the crack tip, I'm pretty sure that you will find that the cells are intelligent enough to know there is some fracture there. And how it happens, I don't know. But we are suspecting from our side, and I'm closely cooperating with someone on this subject, is that we are suspecting that piezoelectricity may be an important way. Streaming potential and diffusion potential and Donon potential may be important as well. But quite often, piezoelectricity is put aside because they say, oh yes, that's too small, etc. We have the feeling that it might not be, that it might be actually one of the methods through which the cells at a distance know that there is something happening and something has to happen, and that upregulating takes place thanks to the fact that they know, oh, there is a fracture there, or there is some inflammation there, or whatever. And of all of the mechanotransduction that we know, I think we know a lot, but I think we don't know everything. And one of the pathways we are testing is the voltage-gated ion channels and electrical phenomena. Because electrical mechanical is so intimately linked in the extracellular matrix that we think that this line of study should be explored. We can show that we've shown in DISC that voltage-gated ion channel responds to load. That definitely, without any doubt, we have clear evidence that voltage-gated ion channels respond to load. The pathway through which it happens is still unclear. But I think it is worthwhile doing. The trouble you may have is that you cannot exactly predict where the crack is going to go. But if you have a controlled experiment, yes, you could do it. Good idea.
Thank you.
Okay.
Okay, thank you very much, Jack, and all of you. With this, we close this session of the morning. And let's go eat.
Now that you know the campus,